Alright, so oh, do you have an English lesson at 10:15, 11:30?
Mm. That a.m.
Okay, so maybe it will have to be 9 a.m. Holy moly.
Oh God.
Time to fix our sleep schedules.
That's not good. I can barely wake up for stats.
Fix your sleep schedules.
Wait, is
I
Miss
Yeah.
Law gonna start teaching
I
at
have
all?
I have no clue. She hasn't told me
Or
anything.
cause like all the other teachers have started.
Exactly. So I'm thinking that she will probably start doing her lessons eventually. Probably by next
Oh,
week,
okay.
she'll probably reach out to you guys. She hasn't
Have
told me
you
anything though.
asked her? Or...
I can ask her. Alright, so yeah, I already started recording actually, so how about we just get into it? Okay, so we're gonna do DNA sequencing today. There are two main topics today. Oh, I guess there's no overview, so we'll just get right into it. So the first thing we're gonna learn is the Human Genome Project. What is a genome? A genome is an organism's complete set of DNA. So all the base pairs. The human genome has about 3 billion base pairs, and the Human Genome Project is an international collaborative research effort that lasted in about 5 years to sequence and map all genes in human beings, or Homo sapiens. So it was actually a race. So most of the sequencing was performed in university and research centers in the USA, UK, France, Germany, Japan, and China. They used a method called small-scale shotgun sequencing, but at the same time, another company called Celera Genomics, so this was a private company, was also trying to sequence the entire genome using shotgun sequencing. So small-scale shotgun sequencing and just shotgun sequencing. So the difference between the university and research centers was that once they had sequenced the entire genome, they were going to make it open, public domain. But Celera Genomics was trying to, uh, they were trying to sequence and map all genes and then make it private. So they were going to try and profit off of the human genome that they had sequenced. So that's why it was a race. There are videos to basically compare and contrast the two methods that they used. So I guess we could look at them, but it it doesn't make too much sense for me to like show the video in my stream. So I think the best option is to just for you guys to watch the videos separately. If you have any questions about the differences between small scale shotgun sequencing and shotgun sequencing, you can ask, but I think the best option is for you guys to look at the videos. I can go over just by word the difference between small scale shotgun sequencing and shotgun sequencing. Actually, yeah, maybe we'll watch the video together. It
Shotgun
it,
sequencing is
it
the
won't
method that
it won't take too long. Or do you guys want to watch the video if I send you guys a link? Does that make more sense? Because
It would be nice to.
they're just a
It'd
minute
be nice to
each.
watch it right now.
Yeah, let's watch it right now together
has represent
and yeah. It's just two videos. So if you guys could watch the private and then the public projects separately in like 2 minutes, and then yeah, and then after that we can talk about it, I guess. Here, I'll watch them at the same time.
was used by the private genome project. Shotgun sequencing requires multiple copies of the genome.
Do you guys just want to watch it in the stream together? Is that a better idea? And then I'll just stream the window.
Sure.
I just wondered like if the quality will be okay.
Sure.
Is it like super blocky?
Hmm.
Shotgun sequencing
Can
is
you
the
guys
method that
see
was
in here?
used by the
Yeah, it's fine.
Alright. Okay, well,
Private Genome Project.
just a minute.
Shotgun sequencing requires multiple copies of the genome, which are effectively blown up into millions of small fragments. Each fragment is then sequenced. The small fragments are assembled using an immense amount of computer power to match overlapping sections. The drawback of this method comes when dealing with repeat sequences. Often there is no way of knowing how long the repeat sequence is, or in which of the many different possible positions the fragments overlap. Even the incredibly powerful software used to shotgun sequence the human genome couldn't cope with this. So Celera, the private company which relied on this approach, had to use the public data to fill in the gaps left by the repeats.
So that's it. And then we'll watch this one. This is a little bit longer. Small scale shotgun sequencing is a little bit more complex.
As represented by this huge stack of paper, the human genome contains more than 3 billion nucleotides, or DNA letters. The first stage of the public Human Genome Project focused on identifying marker sequences, or unique tags, shown here in yellow, at regular intervals throughout this book of life. Once enough sequences were tagged, various blocks of the genome were allocated to different academic centers for sequencing. To begin the sequencing process, several copies of a section of DNA, represented here as a page of text, are cleaved to produce smaller fragments. Although it looks fairly orderly, this step is small-scale shotgun, which creates numerous random fragments. Each fragment is sequenced. Then computer programs align the overlap between fragments to build up an entire page. Marker sequences, shown in yellow, help establish the order of pages in the Book of Life. This methodical process produced huge amounts of data that have been used to virtually reassemble our genome. However, there are gaps. Repeat sequences are common in the human genome, so repeats from entirely different chromosome regions may be erroneously joined together. It will take many years to detect mismatches caused by the repeat sequences. Some regions, especially near the centromeres, may never be fully finished.
Yeah, that's it. So the biggest difference between shotgun sequencing and small scale shotgun sequencing are those like bookmarks, pretty much. Where the markers at regular intervals in the genome, the existence of those markers and the fact that we know what order that those markers come in, that was the biggest difference between small scale shotgun and shotgun sequencing. Any questions? Alright, cool. Let's move on. Alright, so this is another thing we're going to be learning today. So after the Human Genome Project, we're going to be learning about Sanger method sequencing. So he developed, or Frederick Sanger, I don't know how to pronounce it. I'll call it Sanger. Yeah, okay. He developed something called dideoxy termination sequencing.
Alex, I don't think you're streaming right now.
Oh, I'm not streaming right now? Aha, thank you. There we go. Alright. Frederick Sanger. So in 1975, he developed dideoxy termination sequencing. And in 1980, he actually received the Nobel Prize in Chemistry for his sequencing method. And he also received the 1958 Nobel Prize in Chemistry for his work on the structure of insulin. So, pretty smart dude, won 2 Nobel Prizes. So what is a dideoxyribonucleotide? or a DDNTP. Di is 2, deoxy is, removed oxygen. So dideoxy-NTPs have oxygen missing at both the 2' and 3' position carbons. So in a normal deoxy-NTP, so a DNA nucleotide or a DNA like it's not a nucleotide because it still has 3 phosphate groups, but a building block for DNA, it would still have this 3' OH to continue building. But on a DDNTP, there is no oxygen here. And this has unique consequences. So, what happens during DNA elongation if you use a dideoxynucleotide triphosphate instead of a normal deoxynucleotide triphosphate? Any hypotheses? Yes, Nathan said you can't attach another nucleotide because there is no 3' OH, and that is exactly what happens. If you use a DDNTP to continue your DNA chain, then this DDNTP will be the last base pair. So yes, the 3' OH is needed to react with the phosphate group on the 5' end of the next nucleotide to form a phosphodiester bond. So that can't happen. So when a DDNTP is randomly, so by chance, incorporated into the newly synthesized strand of DNA, the synthesis stops there. And this is the idea that Sanger sequencing is based off of. So what materials is used? Basically, everything that normal DNA elongation uses, or DNA elongation uses, plus these dideoxy-NTPs. So you have a DNAP, you have primers, template DNA, and normal DNTPs, but now you also have these DDNTPs. So this is how it's set up. Each test tube has normal deoxy A, G, C, and T. But in addition to these normal DNTPs, each test tube also has one of the 4 DDNTPs. So if you look here, tube A has normal D GTP, DTTP, and DCTP, and it also has normal deoxy ATP, but it also has, you see here, 10% of DDATP, and this DATP number is also reduced. So what would happen in tube A? If you allow normal DNA replication to occur in the presence of both deoxy ATP and dideoxy ATP, what do you think might happen? Any guesses?
I guess your nascent strand would just stop at one point before, because there wouldn't be any place for a new nucleotide to attach, so it would just be like unfinished.
Cool, that's true. And also that it will stop randomly. That's exactly the answer I was looking for, but also it's the fact that if so, let's say you have this primer and then it continues, continues. There's an A here. This is a possible stop point. This is a possible stop point, and this last A is also a possible stop point. So in this tube A, if you let replication happen, you'll have some fragments that I guess if the primers, I don't know how the primers work, but I'm guessing the primers are all just normal. So if you just allow replication to happen as normal, you'll have some fragments that are this long up to this first A. You'll have some fragments that are this long up to the second A, and you'll have some fragments that continue until basically the last A. Does that make sense? So you have fragments of different length. Does it show it? Oh, my bad, my bad. Since it's A, oh, that's right. Oh my gosh. Big facepalm. It'll stop at a T because the corresponding base that A would bind to is T. So it would have fragments that would stop at this first T, second T, third T, or fourth T. Sorry about that. It's not at the adenine on the template it would stop, it would be at the thymines on the template that it would stop, because then on the complementary, you would have to use an adenine. Big face palm. Yes. So the fragment can be 8 base pairs long, or 11 base pairs long, 16, or 18, all the way to the... I guess you could even go past 18 theoretically to the next T, but assuming this is how long it is, yeah. Cool. So imagine you do that for each of the 4 base pairs. So the newly synthesized strands of DNA have different lengths depending on when a ddNTP was incorporated into the strand. So this process is repeated 4 times with different ddNTPs. So if you look here, it's just a little chart for the other tubes. So just following by the example of tube A, you would have like a 15% DGTP and then a 10% DDGTP. Same thing for tube T and tube C. And that's really interesting because each reaction vessel only has one of the 4 possible ddNTPs. So let's say that you take these 4 tubes and all the different DNA fragments that are in them, and then you visualize them on a gel electrophoresis onto 4 different lanes, one for each ddNTP reaction. So let's look at here. Can you guys see the gel electrophoresis? So if you run these different solutions with the different DNA fragment lengths, then theoretically you could know the sequence of the DNA fragments. But how? Because you can interpret the gel electrophoresis and read it so that you know which base pairs are in what position. So in order to know what side is 5' and what side is 3', what information would you have to know about this gel? What's the biggest thing you would have to know about the gel to know which direction DNA was getting longer at, essentially?
Um, the more base pairs, the like less it travels.
Exactly. So you would have to know which side is the positive. I'm thinking that the bottom here, the gel is moving downwards. So if the gel was moving downwards, which side is the 5' end of the DNA and which side would be the 3' end?
The 5' is at the bottom
Okay, and what made you think that the 5' was at the bottom?
because they move like faster, so they're shorter fragments, and that means that they're closer to the like end or beginning. I don't know.
Okay, yeah, you have the answer exactly. So, let's say I started building my DNA at this 5' end, and this is a 3' end. I say that all the slower moving DNA fragments, which are also longer, is my 3' end of DNA because if I'm adding to the 3' end always, and it's the 3' end that would be longer. Or if you keep adding to the 3' end, then the fragments that are closer to the 3' end are longer. So this is the answer. So once you know that, you can actually find the sequence of your DNA with the 5' and 3' end. Why? Because if you know this column number 1, row or lane number 1,
Battery low.
only has sequences that would basically end at an adenine, then you can say that any time that there's a band here, it's an adenine on your actual DNA strand.
Charging battery.
And then the same thing goes for C's, and the same thing goes for G's, and the same thing goes for T's. But how? How do you know that? Well, okay, so the way you know, or the reason why you could basically conclude this, is because it would have to have stopped at a C. So let's look at this first band, for example. All the bands in this lane, you know, basically stopped elongation at a cytosine. So you know that the most recent strand, or sorry, the most recent nucleotide added to the DNA fragment will be a cytosine. So just based on that, you just start building the strand. So most recent added, adenine, then a cytosine, thymine, guanine, and then you just add that, and then eventually you sequence your entire DNA. So yeah, and this is an example. These are examples of real sequencing gels. Pretty cool. Do you guys have any questions? Any confusion about Sanger sequencing? Do you guys understand how it works? Maybe I haven't given the
Um,
best explanation.
no, it's good.
The percents and the steps. I'm not sure if those are the actual percents. I never really like tried to memorize the percents because I feel like it was just there. The percents were just there to show you that all the different DNTPs are present. It's just that one specific base pair, you'll have some that's deoxy and some that's dideoxy to give you a chance of getting a dideoxy or a deoxy. I think that's basically what these percents are here for the sake of understanding why you would include DATPs and DDATPs. I don't remember having to know the percents exactly. So, today in modern sequencing, the theory is exactly the same as Sanger sequencing, but the method is a little bit simplified. So what happens today is four differently colored fluorescent dyes are used to label the four different ddNTPs in a single test tube. So then all four fluorescent ddNTPs can then be run on one lane on a gel electrophoresis. So how does modern sequencing work? So let's say we have all these DNA fragments. These are all different lengths. So if you ran them on a gel, they would create bands. But the difference here is that instead of having all the different DDNTPs run in separate lanes, we know which DDNTP was binded last because the only DDNTP present has a fluorescent dye attached to it. So if thymine is always red, guanine is always black, adenine is always green, and cytosine is blue, what they do is they basically detect the or light or the fluorescence given off at that point or at that band to tell you what ddNTP was added to the DNA fragment at this band. Does that make sense? So they know that this last band is a thymine because when they detect the fluorescence, it's red, and they know that all the ddNTPs of thymine were tagged with a red fluorescent dye. And the same thing goes for guanine, cytosine, and adenine. So it's a little bit simplified. You only need to run one lane. But a computer would interpret this. So fragments are still separated by size, but they actually show up as colored bands now instead of four different lanes. And colors have different wavelengths, which can be read by a computer, and then the computer translates the colors into the order of the nucleotides. So this is like an example of modern sequencing. I'm guessing the numbers are just the base pair number. Base pair 200, and then 360.
Could you explain how the DDNTPs are colored differently? Like, is something added to them, or do they
Yes.
naturally have a different color?
So the four different DDNTPs are dyed. They're a
How
fluorescent
does that
dye is
work?
added to them. So my best guess would be that like you know how before they used like intercalating agents to stain the DNA. My best guess is they bind something to the DDNTP that allows it to give off a fluorescence. That'd be my
all
guess,
right.
my best guess, that they modify a DDNTP somehow to be fluorescent.
Okay.
So, given that the sequence in the diagram below is read 5 prime to 3 prime left to right. So this is 5 prime, this is 3 prime. If we had this on a gel and this was interpreted from a gel, what is the positive end of the gel? So Nathan is saying left. This is the positive side. Any other guesses? Does
I also
everybody?
think left.
Yeah. So, how did you guys think it was the left?
The left is for the shorter strands, and the strand grows from 5 prime to 3 prime. So, like the 5 prime end is going to be corresponding to shorter strands. I don't. Yeah.
Yeah, that's the answer. Exactly. So, if all the DNA fragments are moving towards the positive end and the shorter fragments would move faster, then you know that the shorter fragments would be towards the positive end and the longer fragments would be toward the negative end. Normally you would get a little activity to do here. Um, but we don't have, we're not going to be doing this activity. Hopefully you guys understand DNA sequencing. This is pretty cool, and I guess it's important because sequencing will come up a couple more times. Any questions? In terms of evaluation, the main things you'll be asked to do is you'll be given this like 4-lane gel, and you'll be asked to interpret this gel into a DNA sequence. So you'll have to say 5 prime, 3 prime, and add in all the bases in between. And then you'll also be given a gel like this, and then you'll be told like, let's say, oh, there's an arrow here. So then you'll be asked to say, what end is the positive end, what side is the negative end, and you'll have to label 5 prime and 3 prime on something like a 4-lane gel like this. Cool. Yeah, that's it for today's lesson, guys. Nice and short. Yeah, if you guys don't have any other questions, that's it.
Okay.
All right.
Thank you.
Thanks, Alex.
No problem.
Okay. Bye.
Oh, you have a question? Yep, ask away. Okay, from an old lesson, yeah. Okay, so intercalating agents. Okay, you want me to? I can bring up the slides too. Um, what slide was it again? Tools? Was it in DNA tools or was it in mutations and repair? Okay, let me bring it up. No, it's way, way up. Here? Yep. Okay, so Nathan is asking if an intercalating agent is fit into the structure or if it is bonded somehow and incorporated. Um, it is not bonded, to the best of my knowledge. It is literally fit into the structure. It's like wedged in between. The best way that I understood it is that this intercalating agent wedges itself between the nitrogenous bases. I'm sure some kind of interaction happens, but I don't think any kind of covalent bonding happens. Okay, so for copying an extra base pair, Would that mean that there's some base pair that hydrogen bonds to it when replicating? Hmm, like the other mutagen. That's a good question. I I wonder if the result. That's a good question. So it's like, let's say you have your normal DNA like this, and then these are the base pairs, and then you have like an intercalating agent here, and DNAP comes along. Okay, normal base, normal base, normal base, normal base, normal base, and then it stutters here and adds an oops and adds an extra base like this. Oh, that's a good question. Maybe ask Miss Law that question because then like the DNA itself would have to like squish to incorporate that extra base pair.
Also, you mentioned intercalating agents when you were referring to the DDNTPs. Could you explain how they were related again?
Oh, so the reason I mentioned it is because remember how they use intercalating agents to stain DNA?
Um,
Intercalating agents are
could you?
not directly related to DDNTPs. I just brought it up because in the modern sequencing method, oh, here we go. In the modern sequencing method, they stain, they don't stain, but they dye the DDNTPs using fluorescent dyes, and they're not related, intercalating agents and fluorescent dyes. It's just that both of them end up allowing you to see the, basically see the bars. They give the bars color.
So how are intercalating agents used for staining?
So oh, I keep closing it. Intercalating agents are used to stain gel electrophoresis. So essentially, let's say I use something called ethidium bromide. What you'll do is ethidium bromide, when you introduce it to DNA, it is an intercalating agent, so it'll go between the base pairs, and basically, I don't know if ethidium bromide is visible under like UV light or something like that, but let's say ethidium bromide is visible under UV light.
Okay.
If you introduce it and it intercalates itself into the DNA strand, then let's say I had my gel where before it was not visible, even under UV light or even to the naked eye. If I if ethidium bromide is present and then I expose my gel electrophoresis to UV light, then the different bands will actually show up under UV light like this. Does that
But
make
isn't
sense?
that part? Isn't that like destroying the DNA? Because like if it were to copy, then it would copy incorrectly.
So, um, all DNA in this like gel electrophoresis is already cut. It's already fragmented. Does that make sense?
Yeah.
So the best answer I could give you is that the DNA here is probably not going to be used for replication anyway. You need
All
to
right.
have some method of viewing the gel.
All right. All right. All right.
Yeah, so Nathan, for your question, I'm not too sure. You should ask Miss Law as to how this extra base pair gets there. If it's a situation like this where the mutagen is somehow hydrogen bonding and interacting with this base pair, or if something else happens. That's a good question. Right, cool.
Thanks,
Okay,
Alex.
no problem.
Have a good day.
You too.